Welcome to the second part of in situ bioremediation and bioaugmentation. We're going to carry on our discussion of the microbial community, an area that has developed quite strongly over the last 10 years, I would say. I'm not a microbiologist. I really don't know much about this subject. I do talk to microbiologists, and they advise me and offer suggestions, and usually very useful. Number of references that I found useful. Well, why would we bother with microbiologists? Why do we need to consider the microbial community? Isn't the fact that the chemical disappears evidence enough that it's working? Well, maybe not. It can disappear because of dispersive dilution. It might disappear because of a chemical reaction. It may disappear because of something we're not doing. So if you paid for the manipulation, you might want to be sure that the manipulation worked. You're manipulating a microbial population. Did the population respond as it should? And again, Pedro Alvarez makes the case that it is important to gain at least some background information to say that yes, the microbial population is responding. And again, combining the microbial and geochemical information provides a much stronger case for regulators or clients that this is in fact occurring. So one of the problems is, how do we put together the questions that we as hydrogeologists have with the techniques and capabilities that the microbiologists have? Again, this communication is often constrained or difficult. So there's a number of questions we would ask. How many organisms are there? Who's there? What can they degrade? Most importantly, what actually are they degrading? And how active are they, and what are they up to? Are they just performing the function we're after? Are they doing other things? Those are typical questions we have, and until perhaps 10 years ago, getting any sort of answer was very difficult. Now I think there's information from the microbial community becoming available that's helping us out here. So let's look at some of these answers to these questions. So again, my very simplistic view of a cell, it's got some DNA in it. Ribosome is in the inside. There's a cell membrane of some kind, and there's interaction across that cell membrane with the environment. We need enzymes to biodegrade contaminants, and those are coded for in the genes. However, simply having the right genes doesn't solve the problem. The enzyme synthesis requires transcription, that is, a transfer of the information from the DNA through messenger RNAs, and carry them to the site of synthesis within the cell. And it needs translation then. The RNA has to code for and start building the specific enzyme that's required. So it's not simply a matter of genetics, it's a matter of having the whole sequence in place. Once it's in place, then we can be confident that the microbial system is actually carrying out the reaction that they've developed the enzymes to do. So let's go through these questions and see what the nature of these targets and information gained is. So a typical question we might have is, how many organisms are there present? One way to approach it, the old way, is a culture-based technique. And typically you get total viable bacteria or most probable number of bacteria. Essentially what this involves is plating a sample of water on a specific growth medium and counting the number of colonies that form. Presumption is that each colony represents a microbe that's active in that system. Well, the problem is, what media do we use? Different media get different answers. Turns out that most bacteria are in fact viable, they're active, but they're not cultivatable. That is, they don't respond to this test. So we often get responses, but we're not really sure that it's the real response that we're after. And so the results are generally poor and at best qualitative. There are a number of culture-independent techniques. The most common one that I've seen is the phospholipid fatty acid analysis. These chemicals aren't stored in dead cells, so they really are a fairly good measure of viable cells, not dead cells. In some cases, we can say something about the nature of the population by the nature of the fatty acids present. There are really no standards for this, so again, it's somewhat difficult to assess the direct numbers, but we certainly can assess whether the numbers are going up or down. So who is there? I don't really care very much who is there. I don't need to put a name to them. I'm more interested in the next question, what are they doing? But many people like to know that we can identify what actors are dominant. So a typical approach is to focus on conservative 16S RNA sequences in bacteria. We have a fairly good map of these sequences in different bacteria, so we could do matching and say, well, it's a Dihelicocoides or it's a bacteria similar to an sulfobacter, something like that. And we can amplify and carry out this analysis through a newer technique called PCR, which we'll talk about later. Unfortunately, we have to have some idea what we're looking for because we're going to use primers that prime for specific sequences. So in general, we can ask if a specific group is there, a group that we can seek, or we can characterize groups in a general sense without being very specific about it. Again, it depends on the primers that we have available and that we choose to employ. So if you're stimulating sulfate reducers, then you probably want to make sure you have primers to identify sulfate reducers being viable. And we can seek the same information on genes that code for the RNA. Both can work. So the next question could be, what can they degrade? Not necessarily what are they degrading, but what they can degrade. What's their capability? We might look at a natural population and wonder if we stimulated by adding nitrate, could the natural population use nitrate for denitrification? So we'd like to know, can they degrade it? So again, we might seek a gene that would support the development of enzymes for that degradation. If the gene's present, it doesn't guarantee that it's going to be expressed, but it suggests that the capability is there. Question that's a little bit closer to the ones I typically have is, what are they degrading? So again, if we detect a specific messenger RNA that it's complementary to the gene function of the degradation we're interested in, then that means the organism is very likely expressing or turning on that specific gene to produce that enzyme. And the more we see of that, it likely corresponds with a higher biodegradation rate. More enzyme being expressed, more enzyme being developed. Messenger RNA is a bit reactive, so it's not a trivial issue to get it out and isolate it from other chemicals in the environment. And if it's being transcripted, it still doesn't guarantee that it's being translated. We still haven't seen the enzyme developed. So there's some uncertainty in this analysis, but we're getting there. So who is active and what are they degrading? That would be, I think, the ideal answer if we could get that. So again, what we'd like to do is target the final product of gene expression, the catabolic enzyme, the enzyme that's going to do that degradation. So we could use enzyme-linked immunoassay techniques that have been developed in other areas of biology and are now coming into the environmental field. Again, the analysis is not easy. How does that analysis normally get done? It gets done by this technique called PCR, polymerase chain reaction. What they do is they amplify the DNA by producing multiple copies of a gene. So this polymerase is capable of replicating DNA. So they can get enough of it so that they can count it. So we need a clean sample of DNA free of other contaminants. We need some primers, nucleoids, various chemicals in some medium. And again, I think there's a bit of a black art in getting that right. And again, we're targeting the final product of gene expression, the catabolic enzyme. In a very quick way, the amplification by PCR looks at the community, extracts the DNA. We then have the total community DNA. We subject it to PCR, and again, by using specific primers, we're likely then not going to get the complete idea of what catabolic enzymes are present. We're going to get some subset of those. So we're going to amplify the ones that we want, or at least ones that are close to the enzymes and RNA that we're after. We then subject that to an analysis. And we can cut out bands that appear in the analysis, clone them, and sequence them, and perhaps get right down to the genus level. Or we could subject them to something called DGGE. denaturing gel electrophoresis, and they separates out the bands of DNA or RNA, and we can compare them to standards or typical genuses or species and say whether or not there's a good match. So we can identify that in fact that bacteria is present and it's active. So if you're stimulating by adding sulfate and you want to show that sulfate reducers are present and active, you might start off by a background sample showing there's relatively few sulfate reducers that are active. Then as you stimulate, you might show that more sulfate reducers are active by these techniques. Again, biochemical techniques that we don't play a part in, but again, it's part of that dialogue with the microbiologist to make sure we're asking reasonable questions and they're providing answers that at least approach answers to the questions we're asking. So there appear to be a number of ways of answering our stupid questions, and that's improving considerably over time. So we should expect better and better answers as time goes on. So that's it for part 2. We'll come back to the more hydrogeological aspects in part 3 of this lecture. Thank you.